Welcome back friends. Next we are going to discuss about the micropropagation, regeneration in vitro pathways and factors controlling regeneration. The plant regeneration involves the in vitro culture of the cells, tissues and organs under defined physical and chemical conditions. Critical for in vitro plant propagation and biotechnology, this phenomenon is also applicable to studies of plant developmental regulatory mechanisms. Differentiated cells are able to regenerate into the full array of tissues under appropriate culture conditions. Plant regeneration can be achieved by organogenesis and somatic embryogenesis. Organogenesis means formation of organs from the cultured explants. The shoot buds or monopolar structures are formed by manipulating the ratio of cytokinin to auxin in the cultures. In somatic embryogenesis, the totipotent cells may undergo embryogenic pathway to form somatic embryos which are grown to regenerate whole plants. It was first established in carrots where bipolar embryos developed from single cells. Here is the picture showing the organogenesis and the somatic embryogenesis. The direct organogenesis also leads to the formation of the plant. Next, the somatic embryogenesis, there are mainly two types. Direct in somatic embryogenesis and the indirect somatic embryogenesis. And here also there is direct organogenesis and indirect organogenesis, which is showing to the formation of the plant. The factors controlling the regeneration. The plant materials is extremely important for the success of the tissue culture. The factors affecting the explant's tissue culture response are the genotype, physiological stage of the donor plant, explant source, explant age, explant size, explant position in the donor plant, and explant density. Plant segments used in tissue culture as explants are stem, root, leaf, flower, ovule, cotyledon, and hypocotyls. Such these explants form direct and indirect organs and embryos. Moreover, shoot tips and meristems may give successful results for the callus formation and the shoot regeneration. The first is the physiological state of the donor plant. Explants show an ability to express totipotency are the most suitable for the tissue culture. Explants should be isolated from healthy plants with high cell division for successful response to tissue culture. On the other hand, regeneration capacity of the mature tissues is quite low. Young tissues and organs have a high regeneration capacity than the older ones. The second is the explant source. The plants grown under the greenhouse conditions give rise to better results than the ones grown in field conditions. There are huge variations regarding tissue culture response in explants excised from the plants grown in field condition depending on weather conditions during the year. Sorry, here spelling is WEATHER. However, the best results are obtained from the explants excised from the in vitro grown seedlings. The explant age, size, position in the donor plant, etc. also should be noted before the culturing. Surface sterilization process. All tissue culture studies which aim to obtain high frequency shoot regeneration, which is also a prerequisite for an effective transformation system, should be performed under sterile conditions. The most important treatment prior to culture initiation is perhaps the surface sterilization of the explant. Since in vitro conditions provide bacteria and fungi with an optimal growth environment, unsuccessful sterilization hinders the progress of the tissue culture studies. The next is the culture medium and the culture condition. The composition of the growth medium is an important factor affecting growth and morphogenesis of plant tissues. Plant tissue culture medium consists of macronutrients, micronutrients, vitamins, amino acids, or other nitrogen supplements, carbon sources, organic supplements, solidifying agents, and growth regulator. The Murashik skook is the most commonly used medium in the plant tissue culture. After explants placed on growth medium for different purposes, they should be cultured in culture rooms where the environmental factors such as temperature and light are controlled. Different species may need different environmental conditions for successful culture. Lighting, lighting in the culture room is realized by fluorescent tubes. Control equipments of tubes should be set up outside the culture room. Otherwise, they may cause overheating inside the room, and in that case, extra cooling is necessary. Due to the light sources are inside the culture room, there should be an efficient cooling system to maintain the constant temperature conditions. So that's all about the micropropagation. Hope you all understood this topic. Thank you.